Now I request our HOD, Dr. Selvi ma'am, to deliver an inauguration or welcome address in this inaugural function. Over to ma'am.
Naren,
Yes,
please
ma'am.
check with the principal sir has come.
Yes, ma'am. Sir is there, ma'am. Sir is there.
Okay. Then we will start. A very good evening and warm welcome to all the participants joined today for the inauguration of webinar series. At the outset, I would like to convey my heartfelt gratitude to our secretary, Dr. C.A. Vasi Madam, a dynamic personality who always encourages to carry out the activities which provide a platform for the overall development of our students. It is my great pleasure to welcome our principal, Dr. M. Lakshmanasamy sir, who has kindly accepted to inaugurate and deliver the presidential address in this webinar series. On behalf of the Department of Biochemistry and my own behalf, I extend a hearty welcome to Dr. R. Sivaprakash, Associate Dean, Research and Development, who has kindly accepted to felicitate the gathering. I am extremely happy to welcome the chief guest, Dr. Ravi Maradasilam, who from Indian Institute of Science Education and Research, ISER, Trivandrum, who is going to deliver the special lecture on hand scrub and puzzle of cellular respiration. And I find immense pleasure in introducing him. He has completed his graduation and post-graduation from Tamil Nadu Agricultural University, Coimbatore, and pursued his PhD program with UGC fellowships in Center for Cellular and Molecular Biology, CCMB, Hyderabad. And the PhD was awarded by Jawaharlal Nehru University, New Delhi. Then he did his postdoctoral fellow in the Department of Plant Biology, University of California, USA, between 2008 and 12. Since 2013, he has been working as an assistant professor, School of Biology, Indian Institute of Science Education and Research, Thiruvananthapuram, Kerala. He is the recipient of several awards, honors, and fellowships. During his graduation, he received Satyavan Kobalapillai Award of TNAU for distinction in agricultural botany and Dr. Clement Williams Memorial Prize for distinction in dairy farming during 1998. During his post-graduation, he received TNAU Merit Scholarship, then Senior Research Fellowship from Rockefeller Foundation's International Rice Biotechnology Program in the year 1999 to 2000. Then he received Baby K. Devarajan Prize for Best Student in MSc and Dr. K. Ramaya Award of TNAU for the Best Student in Plant Breeding and Genetics. He also qualified ARS NET. He has received UGC, JRF and SRF for his PhD. He also received travel grant from DST in 2007. He also received ISDB 2004 prize for best poster presentation during the annual meeting of the Indian Society of Developmental Biologists. And during his postdoctoral fellow, he received ASPB Early Career Award by American Society of Plant Biologists in the year 2011 and University of California Davis Award for Excellence in Postdoctoral Research in the year 2011. Then he received Ramalinga Swamy Re-Entry Fellowship by Department of Biotechnology, Government of India. He is also the recipient of DuPont Young Professor Award in the year 2014 and G.D. Naidu Award in the year 2016. He has 19 research publications, including Nature. He has 3 US patents to his credit. With this introduction, I extend a warm welcome to our chief guest, Dr. Ravi Maradasilam. Next, I'm happy to welcome the faculty members and students from our own institution and other institutions. We all understand the importance of science and technology and innovation in our day-to-day lives and the ways in which they are transforming the world. As this event is an international webinar lecture series, the Department of Biochemistry has planned to conduct the webinar consistently for the students, a platform to gain knowledge in the field of biological sciences and to identify the thrust areas of research and decide where to land in future. Before we begin this webinar, I would like to express my heartfelt thanks to all the speakers of this lecture series who all have shown interest in disseminating their knowledge to the student community. Once again, I welcome you all for this webinar lecture series. Thank you.
Thank you very much, ma'am. Thank you. Sir, as our ma'am mentioned that about lecture series, so I just wanted to remind you, everyone, today Dr. Ravi Murdachalam going to talk about hand scrub and puzzle of cellular respiration. And tomorrow morning, 10 o'clock, will be the Dr. K.B. Rebijit from New Zealand. He is talking about molecular approaches in insect pest management. And on 2nd of December, Dr. Srinivasane Jayaramane is from Malaysia. At 2 o'clock on 2nd, he is talking about nanoparticle as an alternative antibacterial agent. And the last 4th lecture will be on 5th December at 9:30 a.m. by Jagadish Chandra Bose Rajendran. He is from Masonic Medical Institute, New York, USA, on the topic cell-derived vesicles, isolation, functionalization, and biomedical applications. So now I welcome our respected principal, Dr. M. Lakshmana Swamy sir, to deliver a presidential address. Welcome, sir.
Thank you, Narendra Kannan and Sri Madam. Most respected Secretary and Syndicate Member of Bharatiya University, Dr. C. A. Vasiki Madam, Head Department of Biochemistry, Dr. V. Selvi, Dr. R. Sivapraya Sir, Associate Dean R&D, Dr. S. R. Madan Shankar, Dean Academics, and today's our guest, Dr. Ravi Murdachalam, PhD from Trivandrum, Dr. K. Nirbama, Assistant Professor, Department of Biochemistry, faculty members, research scholars, and my dear students. On behalf of the management, welcome you all for International Webinar Lecture Series from 30th November to 5th December 2020. And the topic of today, hand scrubs and puzzle of cellular respiration. Hand scrubs recalled sailing past the white cliffs of Dover during the early morning hours of June 20, 1933. Although he was optimistic about a new life in England, he was experiencing a wrenching forced emigration from his native Germany. As a Jewish scientist, he had been fired from his position as a medical researcher at the University of Freiburg shortly after the Nazis came to power. Like many other German scientists, Krebs quickly fled the country. He spent several years as a refugee, working in temporary research positions before becoming a British. Krebs grew up in Hildesheim, a town that was almost totally destroyed by Allied bombing during World War II. The son of the prominent surgeon, Krebs wanted to follow in his father's footsteps. He entered medical school shortly after the end of the World War I, but soon realized that he was more interested in research than in practicing medicine. This was a serious decision because Germany was in the midst of a crippling economic depression and research positions were hard to find. Fortunately, after completing his medical degree, Krebs was hired to assist one of Germany's leading physiologists, Otto Warburg. When Krebs began studying respiration around 1930, scientists had already been working on the problem for more than a century. By the end of the 17th, scientists knew that the breakdown of sugar, glucose in living organisms was chemically similar to combustion. In both cases, a fuel is burned, oxidized to produce carbon dioxide and water. Throughout the 19th century, physiologists studied this process of organic combustion in whole animal by measuring the oxygen used, the carbon dioxide produced, and the heat generated during respiration. These whole animal studies provide useful information, but with the rise of cell theory, scientists realized that respiration in cell must occur in a series of small steps, each of which releases a tiny amount of energy. Otherwise, the heat given off by burning sugar molecules would destroy the cell. There must be a kind of biochemical pathway leading from glucose through a series of intermediate compounds to the final products of respiration, carbon dioxide and water. Discovering the steps in this biochemical pathway became a major goal for 20th century 20th century biochemists, but they faced serious technical problems. A few years later, Krebs moved to another laboratory at the University of Freiburg, and he began seriously studying cellular respiration in 1932, slowly before he fled Germany. At the time, relatively little was known about the process. Following the lead of Edward Buchner, scientists realized that something similar to fermentation occurred in the cells. A molecule of glucose was always split into two pieces, pyruvate, during the first stage of the breakdown of sugar. This set of reactions, what we now call glycolysis, did not require oxygen. What happened to these glucose fragments during the oxygen-consuming steps of cellular respiration still remain a mystery, although many scientists were working on the problem. So with this introduction, I really appreciate and congratulate the Biochemistry department for your excellent webinar. It's a very research topic, really very useful for the PG and research scholars. Once again, on behalf of the management, I welcome you all for this international webinar series. Thank you for the opportunity given to me. I wish the program a grand success. Thank you.
Thank you so much, sir, for your nice presentation on a presidential address. Now I invite our Associate Dean, Dr. R. Sivaprakash, Commerce Science College. Professor Sivaprakash Athanavelu, sir, is an extensive research experience in microbiology, immunology, and molecular biology, involving cloning of genes and expression of proteins in bacteria and mammalian cell cultures. His expertise in aseptic techniques, microbial identification, manipulation and preservation of industrially and medically useful microbial organisms, and development of immunoassays for pathogen and antibody detection. Basic working knowledge of computer software, as well as he did his PhD in University of Madras, Department of Microbiology, and he did his MSc in Faculty of Medicine, University of Madras. Sai has worked in having two patents, three patents, and Sir has a teaching experience, and he was worked in Melbourne Institute of Medical Sciences, and he was the faculty in Computer Science College in 96 to 97, as well as he was a chief biochemist in Salem Microbes Private Limited, and before that, Sir was working in senior scientist in St. Louis, USA, and the Michigan Nanotechnology Institute for Medicine and Biological Sciences, University of Michigan, USA. Now, welcome, sir, to deliver your felicitation in this grand occasion. Over to Professor Athanavelu, sir.
Sir, I think it is muted.
Hello.
So yes.
Yeah, now it's
Yes.
Okay. Sorry about the small glitch. Warm good evening to our respected dignitaries, learned colleagues, and students who have joined this evening for this online webinar series. Well, it's my pleasure to be talking among this group, and I wholeheartedly wish that all of you will have a fruitful learning experience in this webinar series. And at this moment, like now, I would like to thank our Department of Biochemistry, who are not selfish to select all their topics in biochemistry alone. And if you look at the various titles, it's like, one is in biochemistry, another is in molecular biology or entomology, and another one is like nanotechnology and cell biology. So this is a vast array of titles, and I'm definitely sure that everyone will be benefited by this webinar. Uh, we all think like, we have learned so many things during our lifetime. But if you look back, today's talk by Dr. Ravi Maradachalam, it's on hand scrub and the puzzles of cellular respiration. I was just going through the previous literatures, and I found that his hand scripts publication, for the first time was rejected by Nature journal. So this teaches us, how we should keep pursuing research and not leave it in the middle, getting frustrated. And also that two other scientists, Noop and Maritas, they were the ones who elucidated almost majority of the citric acid cycle steps. So a collaborative environment is always needed for the development of research. Well, I hope that everyone will get benefit from this webinar, and I wish good luck for all the participants and a fruitful learning. Thank you.
Thank you so much, sir, for your felicitation. Now I invite our guest, today's guest, Dr. Ravi Maradachalam, is a faculty, School of Biology, Indian Institute of Science, Education and Research, Trivandrum, and is on the topic of hand scrub and puzzle of cellular respiration. Over to Dr. Ravi Maradachalam.
Yeah. So at the outset, I sincerely apologize for the delay because due to some unavoidable circumstances, I have to reschedule it. So thank you for your patience, and I also thank Dr. Selvi ma'am and also the Kongunadu Arts and Science College for giving me an opportunity. So I think Selvi ma'am forgot to mention that I also studied a semester in Kongunadu Arts and Science College before I moved on to TNAU for my bachelor's in 1993. So I am also an alumnus of Kongunadu Arts and Science College in a way, even though I studied just for one semester. Like when Selvi ma'am requested me to give a webinar, so I thought I will talk about my own research. But then I decided that, okay, since it is biochemistry department, and then I thought that, okay, it is an apt to discuss about the topic which I'm going to present today, hand scrub and the puzzle of cellular respiration. And most of the contents which I want to cover has been broadly touched by both Dr. Siva Prakash sir and also the principal sir. But anyway, I will cover the ones which in detail about why I've chosen this topic. So basically this, I hope you can see the slides, right?
Yes.
Yeah. So this talk is inspired by this book, which is called Doing Biology. Okay. So it's a fantastic book, which contains 7 historical cases in a guided inquiry format, aimed to address several proposals for reforming science education. Why? Because textbooks teach biological content, but however, it is important that the students should learn about the history and nature of science, about science in practice, about doing biology. So every biological fact has a story behind it. So the story helps us to understand how the fact is justified. So it also tells us how science is done. And if we do not know this, we remain ignorant of how scientists work and how important scientific discoveries have been made. So Krebs' own discovery of the metabolic cycle that bears his name is an excellent example of scientific problem solving. It illustrates the creative process by which our knowledge of the living world is generated and the critical process by which scientific claims are evaluated. So like many other case studies, the discovery of Krebs cycle is a compelling story of personal triumph over adversity. So as we can go along, we can see that defying several odds, Krebs was successful in deciphering the important metabolic hub of cellular respiration. Okay, that is Krebs cycle. So I would like to ask students, okay, so other than Krebs cycle, what is the contribution of Krebs to biochemistry? So how many cycles did he discover? Any undergraduates like BS, bachelor's and master students? Please unmute and you can answer. How many cycles do you think Krebs discovered? Any rough guess? So we know Krebs because of Krebs cycle. So any guess, like what are the other metabolic cycles which also bears his name? No one? Okay, we will see like how many cycles, metabolic cycles Krebs discovered in the last part. Okay, so coming to the importance, like, so this is the metabolic chart, okay, so which summarizes the entire repertoire of biochemical reactions that is taking place in our cell. Of course, it may not be visible because there are numerous metabolites. And what is important, you can see from this chart, is the Krebs cycle, which occupies a central place in the metabolic chart. And this metabolic chart was published by International Union of Biochemistry and Molecular Biology in association with the famous biochemist Donald Nicholson from University of Leeds, and Sigma Life Sciences has published this metabolic map. So basically, you can drop a mail to Sigma and they may send it for free, this chart. So generally, this chart I give it to student, like all the students of my class, so that the students don't need to memorize the pathway, so they can refer this chart and they can answer the questions, because it is humanly impossible to remember all the metabolites and all the pathways. Okay, so we generally design problem-based solving, so for which we give this chart. So why I'm showing this chart, because you can see Krebs cycle occupies a central position. So why it occupies a central position, you can now appreciate that Krebs cycle is a central hub of metabolism. What do you mean by central hub of metabolism? So all the catabolic pathways, they converge at Krebs cycle. So what does it mean is that, so many hundreds of different proteins, carbohydrates and lipids, when they are degraded or broken down, okay, so they end up as one or the other intermediates of Krebs cycle. So that is the meaning that all the catabolic pathways, they converge at the Krebs cycle. So in contrast, okay, so all the anabolic pathways, they diverge from Krebs cycle. So what does it mean is that cell uses relatively few biosynthetic precursors, mostly the metabolites of the Krebs cycle, to synthesize vast number of different protein, polysaccharides, lipids, and you name any secondary metabolites. So either they are derived from the primary metabolites of glycolysis or Krebs cycle. Okay, so now you can appreciate why Krebs cycle is a central hub of entire metabolism, because all the catabolic pathway, they converge at Krebs cycle, and all the anabolic pathway, they diverge from Krebs cycle. So having appreciated the importance of Krebs cycle, so you can see Krebs cycle being depicted in majority of the publications. Okay, but so one of the biochemists, okay, Larry Morgan from University of Toronto, okay, so he, in his blog post called Sandbox, okay, so he has mentioned that, okay, I challenge the students in my introductory biochemistry class to find out one single website that correctly shows the reactions of the citric acid cycle with all the correct substrate and products, including water and protons. So what he asked the students is that, he asked the student to show one website that correctly depicts the metabolic reactions of Krebs cycle. So what does it mean is that, so even though the the central hub, like this is a common metabolic hub. So this Krebs cycle is misrepresented or not represented correctly in most of the textbooks and also in the websites. So he says that there are 2 sites that don't count. So what are those sites? One's with images from his book. So he has written a textbook which I have shown here, Principles of Biochemistry, Horton, Moran, and so on and so forth. Okay, so he claims that in his book he has given the correct version of Krebs cycle, and also he says that the Biochemistry and Molecular Biology IUBMB website has the correct versions. So also he gives that in 5 years, so he claims that none of the students have succeeded in showing one website that depicts the correct metabolic sequences of Krebs cycle. So he also challenges, can any sandwalk reader find one? And like the top leading journal, Nature, is not even an exception. Why? Because, so again, Larry Moran, okay, he has taken a picture which I have shown in this diagram, okay, and which is published in this publication, which was published in 2013, and he has just jokingly written, it is time to rewrite the textbooks. Why? Because Nature publishes a new version of the citric acid cycle. So can any of you spot what is the problem in this citric acid cycle that is published or shown in this diagram? So I want the students to interact, because I don't want it to be an one-way traffic where I go on speaking and then you just listen. So I think if it is a two-way interaction, it will be really helpful. So can you spot what is the mistake in this diagram of Krebs cycle that is published by the world's topmost journal, Nature? Students, at least master students. So see, you might have seen studied Krebs cycle in your 12th standard or 11th standard, right? So that's why I'm emphasizing this, because even though the Krebs cycle looks so trivial, but there are a lot of things has to be set right. So that's what I'm just showing this and asking to spot out where the mistake is and what Larry Moran means that, okay, it's time to rewrite the textbooks. Okay, so anyway, the answer I have given in the bottom, like the new version, the version that is published shows that there are 6 NADH molecules produced per one cycle of citric acid cycle. So normally, how much NADH and how much FADH is produced in one round of citric acid cycle? So 3 NADH and 1 FADH is produced in one turn of cycle. So here they have mistakenly shown 6 NADH. So where it is, so the first step where they have shown wrong is the first step where citrate is converted to isocitrate. It's not a oxidation. So basically, again, you can see NAD is not converted to NADH in this step. So this is a wrong step. And again, where is the other wrong step shown? So another wrong step shown is that when fumarate is converted to malate, NAD is not converted to NADH. So this is where it's another thing. Another mistake is also shown is that when succinate is converted to fumarate, it is not NAD. If it is FAD, the flavoprotein FAD acts as the electron acceptor. So FAD is converted to FADH2. So these are the mistakes. So that is why he has sarcastically mentioned that it is time to rewrite the textbooks. Okay, so what is the correct version? So this is the version which I have taken from his textbook, which I've shown earlier. Okay, so without going into the details, okay, so what I want to emphasize here is that you can consider citric acid cycle as a catalyst. So why citric acid cycle can be considered as a catalyst? So what is the property of a catalyst? So you know that a catalyst is a substance, okay, that helps. For example, all the enzymes are catalysts, right? So what the enzymes do, they accelerate the chemical reaction, and thereby it helps in converting the substrate to a product. And in the end result, the enzyme doesn't get biochemically transformed. The enzyme remain as it is. So that is the meaning of the catalyst, right? So in that way, citric acid cycle can be considered as a catalyst. Why? Because can you say why citric acid cycle can be considered as a catalyst? So what starts the citric acid cycle? So oxaloacetate, which is a 4-carbon compound, this is the starting point of the citric acid cycle, which combines with 2-carbon acetyl coenzyme A, and it gives rise to a 6-carbon. So 4 plus 2, it gives rise to 6-carbon citrate. Okay, so what happens at the end of the cycle is that the oxaloacetate, which started, it gets regenerated. So the cycle starts with oxaloacetate, it combines with acetyl-CoA by a Claisen condensation reaction to give rise to 6-carbon citrate. And during the intermediate step of the Krebs cycle, 2-carbon that is contributed by the acetyl-CoA is released as carbon dioxide. And at the end of the reaction, the 6-carbon citrate is converted to oxaloacetate. So basically, we started with oxaloacetate and we ended with oxaloacetate. So thereby, you can consider that the citric acid cycle act as a catalyst, because oxaloacetate, it starts, and then at the end of the reaction, oxaloacetate is regenerated without getting converted to any other form. So what is the end product of cellular respiration is that, so the electrons are extracted in the form of NADH and FAD, and also one substrate level phosphorylation happens where GDP is converted to GTP. Okay, and apart from that, 2-carbon contributed by acetyl-CoA is released as carbon dioxide. Okay, so this is the, in short, what happens in the Krebs cycle. So what is the function of Krebs cycle in in human cells? So the main function of Krebs cycle, irrespective of the cells, is to shuffle carbon skeletons and also to break down the molecules. So all the biomolecules except the nucleotides. Okay, some portion of nucleotides also broken down by Krebs cycle. So measure all the carbohydrates, lipids, proteins has to enter Krebs cycle for its oxidation. Okay, so that is what it, and also so the main function is to shuffle carbon skeletons and also to break down the molecules for generation of energy. So depending upon the tissue, Krebs cycle can function like depending upon the tissue, the Krebs cycle can have multiple uses. So for example, so what is the use of Krebs cycle by liver? So the main function of Krebs cycle in liver is to store glucose for use by other organs. So how it is mediated? So all the Krebs cycle intermediates are diverted for gluconeogenesis. So that means the carbon skeletons from amino acids are converted back to glucose. So that is the main role of TCA cycle in the liver. Okay, apart from that, liver also is involved in shuffling the carbon skeletons from one source to or other into new molecules. Say for example, like for biosynthesis of heme, okay, succinyl-CoA can be diverted. Succinyl-CoA, as you can see, is one of the important metabolites. So succinyl-CoA can be produced from acetyl-CoA, which is the end product of fat lipid metabolism, or degradation of fatty acids. Say for example, glutamate gets converted to alpha ketoglutarate and it can enter Krebs cycle and it can be converted to succinyl-CoA. So in this way, you know, the carbon skeleton can be reshuffled. So this is the main function of TCA cycle in the liver. So what is the function of TCA cycle in the brain? Its the main function is to produce energy from glucose. And what is the main function of TCA cycle in the fat, the lipocytes or adipocytes? So the main function, as you know, it's synthesis of fats and cholesterol and the steroids, right? And so this, so actually, before going into the puzzle of cellular respiration, so how, like having appreciated what is the importance of Krebs cycle, okay, and before going into the main topic, so I would like to show a fantastic video, an animation, which is produced by the Smart Biology. Okay, so you can visualize. So this 3-minute video, it summarizes the entire cellular respiration. Okay, so to begin with, so I will start the video here. Okay, so what is shown is the cellular landscape. Okay, so inside the cell, so what is shown highlighted is the nucleolus. Okay, and then it's surrounded by the nucleus, and you can see chromosomes, and then again the endoplasmic reticulum network is shown, and then again all these like Golgi bodies and lysosomes, and then all the cytoskeletal structures are shown. And again, now again you can see the mitochondria here. So why mitochondria? It is the hub of cellular respiration where Krebs cycle takes place. So now we will zoom into the mitochondria. Okay, so let us, so now we are, this video, it gets zoomed into the mitochondria. So now we will see outside the mitochondria is the cytoplasm. In the cytoplasm, you can see a sequence of 10 steps. So these are the enzymes. So you can see these are different enzymes of glycolysis, which is seen outside the cytoplasm. And you can see there is the metabolic crosstalk. So what you can see, so this is the glycolysis pathway, and this is the outer membrane, and this is the inner membrane, and you can see this is the pyruvate dehydrogenase complex, and this is the entire Krebs cycle. Okay, and this is the cristae in which the electron transport chains are embedded, and what happens is that the electrons extracted from the Krebs cycle are transported via the electron transport chain, and then you can see here the ATP synthesis take place. So as you can see here, so this is the first, so this is the pyruvate dehydrogenase enzyme, which is the biggest enzyme, even bigger than ribosomes. Okay, and what you can see is that, so you can see the beautiful functioning of the pyruvate dehydrogenase enzyme. So you can see there is constant movement back and forth, right? You might have heard, recall the structure of pyruvate dehydrogenase. So there is E1, E2, and E3. Okay, so what is this swinging arm? This swinging arm is a portion of the E2 enzyme, which is the lipamide. So basically, it shuffles the intermediates of the Krebs cycle from one inactive site to the another. So again, here is the workhorse of the Krebs cycle. So all the enzymes of the Krebs cycle is shown in this system as Krebs system, starting from pyruvate dehydrogenase, citrate synthase, aconitase, isocitrate dehydrogenase, and again you can see so the electrons extracted from the NADH, it is now passed to the electron transport chain that happens in the inner mitochondrial membrane, and what you see, the fluorescent dots are nothing but the electrons. So electrons from the NADH and FADH are donated to the the different complexes of the electron transport. Again, you can see here. Okay, so what is moving? This is the mobile electron carrier ubiquinone. Okay, and this is the mobile electron carrier cytochrome C, which is present in the other side of the inner side of the mitochondrial membrane. And this beautiful image shows the proton gradient setup, and then how this proton gradient is utilized for rotating the turbine of the ATP synthase for generating the ATP. So ADP is converted in this. So basically what you see, the fluorescent dots are the electron that are transported. This electron transport creates a proton gradient. As you can see, the protons are moving. Okay, and then these protons, when it gets pumped again, like from the inner mitochondrial membrane via the ATP synthase, then the conformational changes that is caused by the ATP, like the F1 subunit of the ATP synthase helps in the synthesis of ATP. Okay, so this is the overview of cellular respiration. So now we will go on like how Krebs solved the puzzle of cellular respiration. So like most of the history has been given by the principal sir when he introduced the Krebs. So again, I will go over it quickly. So Krebs was born in a Jewish family, and his father was a ENT surgeon. Okay, so as principal sir mentioned, so he followed the footsteps of his father and he studied medicine, and later he got interested in research, and to continue his research, he was hired to assist the leading physiologist Otto Warburg. So what is the contribution of Otto Warburg in biochemistry? So at least I expect some answers from the audience. So he's one of the leading physiologists. And what is the contribution of, so Otto Warburg won Nobel Prize, right? So you can see when we come across all the scientists with whom Krebs had connection, all of them have won the Nobel Prizes. So what Warburg won Nobel Prize for what? So you might have heard about Warburg effect when you study cancer biology, right? So Warburg was important. So actually he worked out basically he got Nobel Prize for his work on cancer cell metabolism. Okay, so I without going into the details, so why Warburg is important, because he was an excellent teacher who set high standards for himself and his assistant. And since Krebs worked as an assistant with Warburg, okay, so he mastered the techniques, especially one of the important contributions of Warburg to biochemistry is, so he perfected one instrument, and this instrument was pivotal in deciphering the Krebs cycle. So can you name what is the instrument? At least students. So what is the instrument? So I think now this instrument, I don't think none of the laboratories has it. But this is a beautiful instrument, which the only one instrument which I think Krebs used to decipher the entire Krebs cycle. Okay, the instrument is nothing but Warburg, it's manometer. We will see in the next slide. So basically, Warburg had a huge influence on Krebs as in his research career. Why? Because one is that he perfected the instrument that Krebs used for discovering for his experiments, and also as a mentor, Warburg claimed that all his accomplishments were partly driven by a desire to emulate him. Okay, so this is the Warburg manometer shown in this diagram. Okay, so and this is the reaction vessel. So in this reaction vessel, so what is the tissue which Krebs used to decipher Krebs cycle? So it is nothing but it is pigeon's breast muscle. Okay, so he took a slice of pigeon's breast muscle and he placed in this reaction vessel. Okay, and this reaction vessel is attached to the Warburg's manometer. Okay, so it is shown here. So basically, as the respiration occurred, okay, oxygen was consumed, and the carbon dioxide produced by the respiration inside this reaction vessel was absorbed by potassium hydroxide, which is placed in the reaction vessel. Okay, so because both oxygen and carbon dioxide were removed, so the air pressure inside the manometer decreased. So as a result, the fluid was drawn upward. So basically, it will be in the equal levels to begin with. And once the respiration take place, then because of the decrease in pressure, there is an increase in the solution level. So thereby, you can measure the change in the height of fluid, measures the rate of respiration. Okay, so this is how, the Krebs studied respiration. Okay, so let us thus, so this Warburg manometer and the entire tissue slice technique were regarded as two most important contributions to the advancement of biochemistry, and also it revealed the true beauty and interconnectedness of metabolic pathways that drive life on Earth. Okay, so this Warburg manometer is unusual. Why? Because it could be used to study respiration in cells rather than in whole animals. Okay, so as I mentioned earlier, thin pieces of animal tissue can be sliced with a razor blade, which is constituted the experimental material, and this survived for several hours to finish physiological experiments. Okay, so now this problem of cellular respiration was existing since the beginning of 20th century. Okay, so more than a century earlier, scientists knew that the breakdown of sugar, that is glucose, was chemically similar to combustion. So physiologists studied this whole process using whole animals. Okay, so but it is very difficult to get into the different organs to measure the cellular respiration. So with the rise of cell theory, as principals have pointed out, cells rather than whole organism become the focus of biochemistry. Okay, so the discovering the steps in the biochemical pathway became a major goal of 20th century biochemists, and how to study the chemical reaction that is happening within a living cell. Okay, if it is a living organism, then it's easy. But within the living organism, there are trillions of cells, and how to get inside the cell to study this respiration. So at that time, so Ed Buckner, as again principals are pointed out, so in 1897, Ed Buckner demonstrated that fermentation could occur outside the cell using cell-free extracts. So what he does is that he extracted the enzymes from the cells and used those cell-free extracts to demonstrate that fermentation can happen outside the living system. So for this discovery, Ed Buckner was awarded the Nobel Prize in Chemistry in 1907. Okay, spurred by this finding, people attempted to study cellular respiration using cell-free extracts. So do you think whether people succeeded? Like fermentation, since it involves only one enzyme, that is lactate dehydrogenase, it's easy. Okay, whereas you know that the Krebs cycle involves several enzymes. So even if you make a cell-free extract, do you think that people will succeed if they use cell-free extract to study Krebs cycle? Yes or no, students? So Ed Buckner succeeded, whether people succeeded or not. The answer is no. Why? So why they cannot succeed? So they can extract all the enzymes in the cell-free medium except one enzyme of the Krebs cycle. So what is that enzyme? So if you study Krebs cycle, so most of the enzymes of the Krebs cycle are located in the matrix of mitochondria except one enzyme that is located in the inner mitochondrial membrane. Right. So what is that enzyme? So that enzyme cannot be extracted in a soluble form from the in the cell-free extract. So because in the absence of that enzyme, so Krebs cycle cannot continue. So what is the enzyme which is located in the inner mitochondrial membrane? Whereas all the enzymes of Krebs cycle, that is only one enzyme that is located in the inner mitochondrial membrane, which is a part of the complex 2. So that is the reason why people were not successful in mimicking what Ed Buckner done for fermentation. So Krebs had an answer to the problem. So he asked that, okay, instead of using whole animals, why can't we use the thin slices of tissue that was used in Warburg's manometer to find the clues? So Krebs' proposal to Warburg was rejected, and Warburg refused him permission to continue this new line of research. Why? Because he was interested only in cancer metabolism, and he doesn't want to deviate from his own research focus, and then thus he forbidden Krebs from studying this aspects of cellular respiration. Thus the discovery of Krebs cycle has to wait until Krebs left Warburg's laboratory and started his own group. Okay, and so even though Krebs regarded Warburg as his mentor, so one of in one of the students of Krebs, Kornberg. Okay, so what is the again? Krebs and Kornberg, they proposed another important metabolic cycle, okay, which is a bypass of Krebs cycle. Okay, so he mentioned that, okay, Warburg in his memory recollected that Warburg made it clear to Krebs that he would not help Krebs obtain a university post once he left the lab, and indeed he sent a letter to Krebs' future employer urging him to keep Krebs busy with clinical duties. Why? Because Krebs is a doctor by profession, so he suggested that he makes a good doctor and he will not make a good researcher, and thus he would never ever make a good biochemist. But ultimately Krebs had the last laugh, because he won the Nobel Prize for discovering the Krebs cycle. Okay, so Krebs in 1931, Krebs moved to medical clinic in University of Freiburg, and there he discovered urea cycle along with his medical intern student Henseleit. Okay, so this is the second cycle. So in fact, this is the first metabolic cycle that is discovered by Krebs, even 5 years even before the discovery of the Krebs cycle. Okay, in 1932, Krebs discovered urea cycle, and this discovery made Krebs to give an international recognition, and he was invited for several lectures, and then for his fame, he was recommended for a tenure at University of Freiburg. Okay, but unfortunately, as again principals have mentioned that, so in the 1993, when Hitler took the charge of Chancellor of Germany, okay, so he ordered all the non-Germans should be dismissed from the professional jobs, and as a result of which, Hans, being a Jew, he was dismissed from his post and forbidden to enter the university premises. So thus he was forced to migrate out of Germany. Okay, so at that time, Frederick Hopkins, again, he also is a Nobel awardee. So what is the contribution of Frederick Hopkins? So why he was awarded Nobel Prize? So he was awarded Nobel Prize along with Christian Eichmann for the discovery of vitamin. Okay, so Frederick Hopkins, so he came to the rescue of Krebs, and he awarded a demonstrated position in the Department of Biochemistry at University of Cambridge. Okay, so Krebs, again, he migrated from University of Freiburg to University of Cambridge. So along with that few personal belongings, he also took like a few couple of manometers that he perfected in Warburg's laboratory, because these are the workhorses. They turned out to be the major equipment in the success of Krebs in discovering the Krebs cycle. So in 1935, he was given a tenured lecturer position in pharmacology in the University of Sheffield, okay, where he continued there for 19 years. So it is in this way, along with his student Johnson, okay, he discovered Krebs cycle. Okay, so Krebs attributed that solving the Krebs cycle is it's like a biochemical puzzle. So why? Because following the lead of Buckner, scientists realized that something similar to fermentation occur in cells. So post-glycolysis, what is the fate of pyruvate remained a mystery. So again, as I mentioned, Krebs, along with the graduate student William Johnson, started to understand the metabolic steps of the oxidation of glucose. So basically, he attributed that solving this problem was like solving the jigsaw puzzle. So he brainstormed with pencil and paper, and he generated numerous hypothetical pathways. So each step in the chemical pathway should obey the rules of organic chemistry. Okay, even though, say for example, you can combine 2 pyruvate to make citrate, but it doesn't obey the rules of organic chemistry, right? So basically, even though you can predict several hypothetical metabolic reactions, but it should always obey the rules of organic chemistry. And it turned out that most of his hypotheses turned out to be wrong, and the only way to validate the hypothesis is to demonstrate by experimental evidence. Okay, so now Krebs started looking for intermediate compounds. So Krebs hypothesized that since burning of glucose is analogous to combustion, so adding fuel to fire increases combustion. Why? Because more oxygen is consumed. So in same analogy, so glucose can be considered as a fresh wood. Okay, so when glucose is burned, okay, so the intermediate compounds, what are the intermediate compounds? Now we know what are the intermediate compounds. They are nothing but the metabolites of the Krebs cycle. So they are the partly burnt ambers. Okay, so what Krebs does is that, so Krebs tried several metabolites. Okay, so say for example, shown in the table is that, so he tried succinate, fumarate, and malate. Okay, and as you can see, when he added succinate, fumarate, and malate, okay, to the pigeon breast muscle tissue in the manometer, you can see these 3 compounds boosted the consumption of oxygen. So from this experiment, Krebs concluded that succinate, fumarate, and malate increase the respiration. However, they still, so again, the puzzle is that, so even though it increased the consume of the oxygen consumption, so they also remain in the reaction vessel. So if it is burnt, then obviously all these 3 metabolites should be completely oxidized to carbon dioxide and water. But that was not the case. So that was a mystery. So how they could be detected in the reaction vessel at the end of the experiment. So that raised 2 questions. So if they were really intermediates in the slow oxidation of glucose to carbon dioxide, why weren't these extra ambers completely burned in the process? Okay, and if they weren't, okay, intermediate compounds, why did respiration increase so much when they were added to the reaction vessel? Okay, so for this, the solution was suggested by Albert Szent-Györgyi. Okay, so again, it's a Nobel awardee. Okay, so he suggested that, so succinate and fumarate and malate were not reactants in the cellular respiration, but acted like catalysts. Okay, accelerating the reactions without being consumed, providing the solution to the first question. Okay, so what he suggested is that, okay, again, please note that, okay, so again, here is the important contribution of Albert Szent-Györgyi. He gave a clue or an inkling to Krebs, suggesting that succinate and fumarate and malate were not the reactions in cellular respiration, but acted like a catalyst. Okay, if true, okay, now you can ask, if it is true, then what are the true reactants? So now we know that the 2 reactants are oxaloacetate and acetyl coenzyme A. But at that time, it was not known. You know that we will come back. So were these 3 compounds the catalyst or reactants? So this controversy remained unresolved for almost 2 years. Okay, so again added to the puzzle is the another intermediate citrate. So the most puzzling piece in the zigzag puzzle of solving the Krebs cycle. Why? Because along with succinate, fumarate, and malate, adding citrate also increased the consumption of oxygen. Okay, so like why? Again, why it was difficult for the biochemist to hypothesize the role of citrate in the later stages of cellular respiration. So why biochemists were puzzled? Because you know that the end of glycolysis, what is the end product? It's a 3-carbon pyruvate. So this 3-carbon pyruvate should enter into the Krebs cycle to get completely oxidized into carbon dioxide in water. But so where is the role of citrate here? Why? Because again, citrate is a 6-carbon compound. So if that is the case, it is possible that 2 pyruvate can combine together to form citrate. So that cannot be possible. Why? Because there is no chemical reaction which can combine 2 pyruvate to form citrate. So again and again, citrate is a 6-carbon compound. So this was a big puzzle to explain what is the importance of this 6-carbon compound in the breaking down of pyruvate, which is the end product of glycolysis. Okay, now we know that the pyruvate enters via link reaction. It combines with oxaloacetate to form the 6-carbon compound. And since the citric acid cycle is a catalyst, it regenerates the oxaloacetate. But it was not known at that time. Okay, so that is the major puzzle. Okay, so at the same time, okay, as again Principal Sir pointed out, so again 2 scientists, Knoop and Martius, okay, so they worked out a linear pathway starting from citric acid to oxaloacetic acid. As you can see, so this is a theoretical pathway, and they published, Knoop and Martius suggested, and they worked out this theoretical pathway which obeys the rules of organic chemistry or chemical principles. It started with citric acid and it ended with oxaloacetate. Okay, so this pathway, as you can appreciate, included all the steps of Krebs cycle in a linear order. Okay, so was this a large missing piece in the biochemical pathway leading from glucose to carbon dioxide? So now what Krebs does is that, so now Krebs try to validate. Okay, so how we have validated whether this Knoop and Martius pathway was correct. So basically, Krebs hypothesis that this linear pathway is like a mountain stream cascading from one pool to another as it flows downstream. Okay, so if it is blocked by a dam, okay, what will happen is that the water will accumulate upstream the dam. Okay, so in same analogy, what Krebs thought is that somehow if you block the linear pathway of Krebs cycle or inhibit the linear pathway of Krebs cycle, so basically what should happen is that the upstream metabolites should accumulate in the pathway. So what he decided is that he chose to block the conversion of succinate to fumarate. Okay, so what he said, so what he found is that when he blocked this conversion of succinate to fumarate, so you can see like all the upstream metabolites, they accumulated when the citrate was added. Okay, so that suggested that this linear pathway is correct. What proposed by Knoop and Martius was correct. Okay, so succinate, again you can see it accumulated even more than even the control. Okay, so now this gave an inkling. Okay, so taking cue from the Knoop and Martius linear pathway and also the proposal by the Alberts and Georgi that it acts as a catalyst. So what Krebs does is that if Knoop and Martius pathway was correct, the intermediate succinate, fumarate, and malate should be completely burned up during slow combustion of glucose. However, they accumulated in the cell. And so what Krebs does is that now if the ends of the Knoop and Martius pathway are connected, okay, several pieces of zigzag puzzle would fall in place. So what is this? So now Krebs hypothesized the cyclic pathway. If a cyclic pathway would explain why the intermediate didn't disappear instead of being completely oxidized. And okay, why? Because if it is cyclic, okay, oxaloacetate instead of getting completely converted to carbon dioxide and water, it gets regenerated, okay, and converted back to citrate. Okay, so what Krebs does is that, so he took the Knoop and Martius linear pathway, okay, and just he tied both the ends. Okay, so he took a linear thread and he made a circular thread. So that is the contribution. So basically, and that he experimentally proved and he validated. Okay, so now everything falls in place. Okay, so what is the important role of citrate? So again, to prove what is the role of citrate, what Krebs does is that he added pyruvate, and he demonstrated that when he added pyruvate, so citrate accumulated, which suggests that somehow pyruvates get converted to citric acid. Okay, so now, but at that time when Krebs discovered or proposed this Krebs cycle, okay, so how pyruvate gets converted to acetyl-CoA is not known. Okay, so this discovery is known 10 years after the proposal of Krebs cycle. Okay, so Krebs now has firmly tied the two ends of the Knoop and Martius pathway together to form what is known as Krebs cycle today. Okay, so as again, Dean mentioned, so when he communicated this finding to journal Nature, so Nature rejected Krebs' paper in 1937, and dejected, stung by the rejection, Nature, Krebs communicated this paper to a Dutch journal, Enzymologica. So they published this, and then again 50 years later, so Nature editor has apologized that it is one of the biggest mistake made by Nature for rejecting the Nature's paper. Again in 2003, another editor considered that it is a notorious rejection. Okay, so although Krebs knew that the pyruvate links glycolysis with citric acid cycle, he did not understand the connection. Okay, so the important role of link reaction, that means how pyruvate gets converted to acetyl-CoA, and then acetyl-CoA combines with oxaloacetate is not known. Okay, so neither he or anyone knew where cellular respiration occurred in the cell. This is another beauty. So they know that cellular respiration occurred in the cell, but again, you know that cell is compartmentalized. So and now we know that this respiratory pathway happens in mitochondria, but when Krebs discovered, it was not known. And again, it was not known what is the importance of ATP at the time when Krebs proposed this cycle. Okay, so how ATP is synthesized in the mitochondria would not be discovered for another 30 years. Okay, so recognizing how many pieces of the puzzle of cellular respiration were still missing in 1937 when Krebs proposed his Krebs cycle, it does not diminish Krebs' accomplishment. Okay, so what this story says is that the scientific knowledge that gets written in textbooks, okay, it is often constructed of small increments. So you see, many of us script that, okay, I have to study Krebs cycle, but you can see how much difficulties and how much painstaking experiments he has taken to decipher this important metabolic hub of the cellular respiration. Okay, so again, this leads to several questions. Why was Krebs able to see a biochemical cycle where other scientists still saw it as visualized as a linear pathway? So again, you can answer, is it a flash of insight as he discovered another biochemical cycle several years before the citric acid cycle? Is just because since he discovered Krebs urea cycle, so whether that insight that made Krebs to tie the ends of the Knoop and Martius pathway. Okay, so again, you can ask questions. What might account for his achievement? Is it careful choosing of the problem, highly structured approach to work, intellectually flexible? Why? Because Krebs quickly abandoned the hypothesis that could not be supported by experimental evidence, so that he is not bogged down in fruitless dead ends. And again, he volunteered reworking the problem from a different perspective, in the sense that it emerged after reading the Knoop and Martius paper. So even so, it took several days of thinking and experimenting before Krebs began to realize that tying the Knoop and Martius linear pathway would produce another biochemical cycle. Okay, so again, as I asked, what are the four metabolic cycles that is associated with Krebs? So one, as I mentioned, the first metabolic cycle discovered by Krebs is Krebs and Henseleit urea cycle, which was proposed in 1932, followed by Krebs-Johnson citric acid cycle, 1937. And again, the third cycle proposed by Krebs is the Kornberg-Krebs glyoxylate cycle, which is again a bypass of Krebs cycle. And the fourth cycle that you won't see in any of the biochemistry textbooks that has been mentioned explicitly is the uric acid cycle. That is, again, maps Krebs uric acid cycle, which was proposed in 1978. And it was, again, many biochemists established the intermediates of purine biosynthesis or purine metabolism is a linear process. It was Maps and Krebs who organized the pathway as a cycle, and the Krebs urea cycle is hidden in this publication, Biochemical Journal. Okay, so again, this has been taken from this biochemistry textbook, Metabolism at a Glance. Okay, so most of the textbooks don't say the fourth cycle. So basically, as you can appreciate, Krebs has been credited with discovery of 4 metabolic cycle. Okay, so this is the urea cycle, which is like most of the textbooks don't appreciate it or give it. Okay, so I'm not going into the details of it. So again, so now I can give some lot of questions to debate. Okay, Krebs called his cycle the tricarboxylic acid cycle and also a citrate cycle after the first intermediate of the TCA cycle. So today most biochemists refer to this process Krebs cycle. So the question for debate is, so does this honorific name give too much credit to one scientist among the many who worked on the problem of cellular respiration? So in other words, how much credit do you think the Krebs deserves for the discovery of Krebs cycle? Whether Knoop and Martius deserve contribution in the discovery of this Krebs cycle? And of course, so one of the important thing hidden in the discovery of Krebs cycle is the contribution of Johnson. Okay, so Johnson, he is a student of, is one of the PhD student of Krebs, and his entire PhD thesis on is centered on deciphering the Krebs cycle. Okay, so although Krebs did a small amount of his own experimental work, most of the data on which the cycle was based was collected by Johnson. Okay, so however, Johnson was by no means just a pair of hands. So even Krebs has confirmed that Johnson could work independently, describing him as being a particularly conscious, reliable, skillful and steady worker who has also shown independence of thought and sound criticism. Okay, but so here again it gives rise to the question whether Krebs cycle should be rightly called as Krebs-Johnson cycle, which some of the textbooks do. Okay, but some of the textbooks don't say it's as a Krebs-Johnson cycle. It's called as Krebs cycle. So it's always important that due credit should be given to the contributors. Okay, so again this can be a debate, like how much credit does Arthur Johnson deserves along with Krebs for this discovery of Krebs cycle. Okay, so again with this I will end this talk. And again, as you can see in this slide, so this is the stadium from the University of Sheffield. Okay, where Krebs worked for 19 years, and as you can see in the stadium, they have painted a bicycle. So this is to represent Krebs bicycle. So I think the students of biochemistry know what is a Krebs bicycle. Okay, even though Krebs discovered 4 metabolic cycles. Okay, so what are the 2 Krebs bicycle, and what is the connecting link between the 2? So one is Krebs cycle and another is urea cycle, and both the thing can be interconnected by arginosuccinate, the metabolite arginosuccinate. Okay, why? Because when the arginosuccinate gets cleaved, so one important metabolite is fumarate and another one is arginine. Okay, so arginine enters the ornithine or urea cycle, and fumarate from the cytoplasm can be converted to malate, and from malate can enter into the mitochondrial matrix to get converted to be a part of the urea cycle. So that's why it is called as Krebs bicycle. Okay, so this University of Sheffield, they have painted to encourage people to start cycling on a regular basis, to highlight the health benefits of the sport, and also it is a dedication for Krebs. Okay, so the local school children there will play the part of molecules in the reenactment of the way the Krebs cycle works on the field. Okay, so with this, I will end my talk, and I can take any questions. Thank you. Sorry, I have to rush because for maintaining the time. So I have to rush. So I am now, I can take any questions and we can discuss.
Yeah. Thank you, Dr. Ravi. Now the session is open for discussion. Please raise your questions and
so so rather than like using the chat box, I think people can unmute and then they can ask the questions.
Please don't leave from the meeting hall because we have to. I will be posting the feedback form as well as attendance form. Please stay here.
Again, a question for students. So how many cycles the Krebs cycle has to run for releasing the two carbon from acetyl-CoA, 2 molecules of carbon dioxide? Is it 1 cycle, 2 cycle, 3 cycle, 4 cycle? Or multiple cycles? So this is one of the questions asked in the competitive exams. So it is often easily overlooked. So how many cycle does Krebs cycle has to run, okay, to release carbon as carbon dioxide? Release carbon from 1 acetyl-CoA molecule as 2 molecules of carbon dioxide. And second question is that Krebs cycle, as we have seen, it runs in the clockwise direction. Can it run in the anti-clockwise direction? And if it runs in anti-clockwise direction, what is the significance? So these are some of the questions to ponder.
The students,
Excellent.
please. Yes. Proceed.
See, I could say
Excellent
around 80 students, but
presentation,
no one
Ravi.
is asking. Yeah.
Yeah, I have a question.
Yes, ma'am.
How many molecules of ATPs are produced when 1 molecule of NAD is getting oxidized through electron
Okay.
transport?
Yeah, that's
Actually,
again.
it is given in the textbook as 3 ATP molecules. Now it is latest research and latest publications show 2.5 molecules of ATPs.
Yes, ma'am. So actually, so the thing is that, for ease of calculation, for example, if you want to calculate how much ATP molecules are produced by oxidizing 1 molecule of glucose. So for ease of oxidation, and previously in the old textbooks, it mentioned it is 3 molecules of ATP. Now I think the refined number is around 2.5. And even people say that it may be even less. The precise number of ATPs is not known clearly, but as a rule of thumb, for working out the bioenergetics, it is recommended, if you want to make the calculation easy, use the number 3, rounded integer 3, if not 2.5. So you can use both the numbers, but still it is not at, so the complete number is not at known.
Okay, thank you.
But it is 2.5 is the latest, as you mentioned, it is the latest estimate, which is widely accepted by the biochemists all over the world.
Okay, even I will be getting confused while.
Yes, ma'am. That is the reason why, ma'am, when we calculate the energetics, so they give a range. Okay, so the range can be somewhere from 28 to 38, so somewhere between 28 to 36 or something. So this range comes why? Because it depends upon which shuttle the cytoplasmic NADH takes. So if it takes malate aspartate shuttle, then you get maximum amount of ATP. If it takes glycerol 3-phosphate shuttle, then it takes 2 ATPs less. So that is one range. And again, if you use 2.5 ATP molecules for calculation, you get one value. And if you use 3 ATP molecules per NADH, then you get a different value. So that is the reason why, on a safer side, most of the textbooks give a range. So whatever you calculate using these 2 values will fall within the range. So we can't give a precise ATP molecules. Obviously, again, it is not 100% efficient in our system. Lot of energy is wasted as heat energy. So obviously, even the number what we calculate theoretically even may be little less.
Okay, thank you, Ravi. Thank you.
See, students,
from students. Yeah.
Narendra, yeah, please, you are asking.
No, that's what I'm expecting questions from students. So students can ask.
See, I asked 2 questions to student. How many cycles does Krebs, how many cycles it should take for Krebs cycle to run to release 2 carbon that enters as acetyl-CoA to be released as carbon dioxide?
Any PG students or
It's
final year
very
UG
easily overlooked.
Yeah.
So many of us think it is in the first cycle. First cycle, no carbon no carbon that from the carbon skeleton of acetyl CoA is released as carbon dioxide. Okay, so what will happen in the second cycle? Is in the second cycle both the carbons will be released, or it is in the third cycle, or in the fourth cycle, or in the fifth cycle, or in the sixth cycle? Students, so I've given the multiple choice questions, so at least you pick none anyway. I've given one is wrong now. 2, 3, 4 and above.
Students are, I think, even they know the answer, they are hesitating to express, I think.
And again, there are beautiful literature to show. Okay, so why again? So again, I'm asking students the question: why citrate should be converted to isocitrate? What is the chemical logic? So what is the principle behind it? So these are some of the questions. If you understand and read, so you know the metabolic path. Working out the metabolic pathway will be very easy. So you might be like, I don't know how, like, definitely, like, it is a tough task for people to teach metabolism because so many enzymes and so many metabolites are involved, and it is humanly impossible to remember all the metabolites of Krebs cycle. But if you not even Krebs cycle, of all the metabolites of the entire metabolic pathway chart, which I've shown in the second slide, right? So, but can you appreciate that, like, starting from the simple trios, if you understand the logic, okay, of what is happening, you can derive the entire metabolic chart. You can derive on your own if you understand the design principles of metabolism. Okay, there is a beautiful review from Nature Chemistry. Okay, so please read. It is titled as Design Principles of Metabolism. Okay, if you understand those design principles, so starting from the simple trioses, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, you can derive all the biochemicals. Okay, it is just like it's like logical mathematical problem solving. Okay, so you can master the metabolic reactions. You no need to memorize the pathway. If you memorize the pathway, it will evaporate within 1 or 2 weeks. Okay, of course you can memorize the pathway for your exams. That is the reason why I don't encourage my students to memorize the pathway. I just give the chart. Okay, but I ask the question such a way that if they understand the concepts only, they can answer the question. So say, for example, I ask this question, okay, so how much cycle it will take? So they have to work it out. If you don't work out, then you can't get the answer. Students?
Yeah, I think they will take these questions even for, and they will work out. I think they will go through and they will find out the answers. I think we can make it as assignments like thing so that the students can work out on it, and we will find out whether, because if anyone is ready to answer, yeah, okay. So I think in that case, no questions are not people ready to answer also. So we can move on to the next session for vote of time. I think, is it okay, Ravi, or have you completed this
No
course?
problem. Yeah, I'm fine with whatever.
Yeah. Okay. So now I call upon Dr. Nirupama, Assistant Professor, Department of Biochemistry, Government Science College, to deliver a vote of thanks. And before the vote of thanks, I would like to tell about tomorrow's program. Yeah, it is by Dr. K.B. Rebijit, Senior Scientist, Molecular Entomology, Ministry for Primary Industries, New Zealand. The topic will be the molecular approaches in insect pest management. But time is at tomorrow 10 o'clock in the morning. So please join by 9:50 a.m. in the morning tomorrow. So now I over to Dr. Nirupama for the vote of time. Thank you, ma'am.
Sir, may I audible, sir?
Yes, ma'am.
Yes, yeah.
Good evening to all. I feel honored and privileged to get opportunity to propose a vote of thanks on this webinar series to those who have directly and indirectly contributed to this event in effective manner. First of all, I thank our Honorable President, Dr. M. Archami, sir, who graced us with his support and guidance. We are extremely thankful to our beloved secretary, Dr. C.A. Vasiki ma'am, to her constant motivation and support behind all our endeavors. Thank you, madam. I also extend my thanks to our principal, Dr. M. Lakshmanaswamy sir, for his continuous encouragement and delivering the presidential address. I would like to thank our Associate Dean and R&D, Dr. R. Sivaprakash sir, for his great support in conducting this event. My heartfelt thanks to our HOD, Dr. V. Selvi, Associate Professor and Head of the Department of Biochemistry, for her constant guidance in organizing the webinar series. I thank our today's resource person, Dr. Ravi Maruthachalam, for his thought-provoking and valuable speech about the unknown facts about hand scrub and contribution in the field of cellular respiration. And we are really enlightened with his knowledge and presence. I extend my thanks to session coordinator, Dr. R.T. Narendrakanan sir, Assistant Professor, Department of Biochemistry. Finally, I would like to take this opportunity to thank all the participants for your cooperation in making the international webinar a resounding success. Once again, I thank you all for your valuable participation. Thank you.
Thank you, ma'am. So I request everyone join tomorrow morning.